Charlotte Dawson. I've just finished my PhD at the School of Tropical Medicine within the Centre for Snakebite Research and Interventions. So I'm going to present some of the data that I gathered during that PhD, where I was part of that was looking at the intraspecific venom variation of the Puffadder bitis arietans. And just a very brief kind of talk about why venom variation is important. So venom is a key evolutionary adaptation for venomous species, and therefore it's subjected to multiple selection pressures. So things like diet, climate, habitat, and the need for defense, these can all affect the venom that is being produced. And this variation has been documented at various levels, so the genus level, species, and intraspecific variation. A really lovely example of this was a 2021 paper that came out earlier this year by Sanjay Laxmi in India. And she showed that there's huge variation in the cobra Naya Naya. And what they also showed was that this can have real clinical implications on the efficacy of antivenom. So in murine models, they found that venoms from the northern part of the country the lethality was not neutralized by clinically available antivenoms. So it's something we need to bear in mind. And bitis arietans it has a huge geographical range across sub-Saharan Africa and into Arabia. And you can see from these two images, they also vary hugely in their appearance. And they're large, heavy-bodied vipers, and they inhabit a huge range of habitats. And this can often bring them into conflict with humans. And because they're so common, they're where people tend to be around, where they're herding. This makes puffaders they're held responsible for the majority of the annual approximately 32,000 snake bites that are occurring in sub-Saharan Africa. And these are very clinically serious bites. They manifest in very rapid, painful swelling, blistering, bruising, necrosis, hematotoxicity. And these can be fatal bites if you don't quickly access antivenom. And the necrosis can often progress to the point where amputations are required. So this then has lifelong implications for snakebite victims. And it's a great species to look at for geographical variation. It has this huge range, and there's already evidence within the literature that there is variation occurring. So there's a 2010 paper by Rachel Currier where she did STS page, which you can see up in the corner here, of 6 different locales of bitis arietans. And you can see they've got variation in the proteins present in their venom. And then more recently in 2018, there was a paper just looking at individuals from across Angola. And as you can see here, just looking at their PLA2 activity, again, you see huge variation in this. And there also does start to appear to be sex-based differences, but I'm just going to focus on geographical here. But what we currently don't have, and the niche we kind of looked at in this project, was transcriptomic comparisons between different bitis arietans individuals, and then taking that further to look at what the variation might have on the efficacy of antivenom. So I was very lucky that we have several geographical locales cared for here at CSRI, so I was able to access the venom from these snakes, and we have venom glands dissected from them. We had one individual per location that we took the glands from and isolated the mRNA and submitted this for sequencing. I used the venom to build an activity profile to see what's going on functionally, and then we looked at 4 medically relevant antivenoms in a couple of different assays. So just as a brief overview of the transcriptomics, you can see looking at these pie charts, we had a huge variation in the makeup of these venoms. So bitis arietans from Nigeria is hugely dominated by C-type lectins. It had the most of the 3 different locations we looked at, whereas comparing this against Tanzanian bitis arietans, you see there's this huge selection of SVMP expression. Again, you see a reasonable component made of C-type lectins. And then finally, the last location I looked at was South Africa. And this was a slightly more complex venom, but again, it's highly rich in transcripts of SVMPs and C-type lectins. But what we are very well aware of is that the protein level seldom reflects a transcriptomic level. There's a lot that can happen between a transcript being made and it becoming a mature protein. There can be changes in an expression level, it can be silenced. So the next thing we wanted to look at was, are these transcriptomic profiles actually reflective of what is going on? So we ran some analytical gel filtration chromatography of 3 different venoms. I will note at this point that these venoms are pooled from the individuals from these regions at CSRI. So that could be a reason for any discrepancies we see. And then Mark Wilkinson, who I work with, is a highly skilled gel chromatography expert, and he was able to interpret these to inform us of proteins. So looking one by one again, so in the Nigerian, we see this high expression of C-type lectins, which we would expect. There is SVMPs present, and we did detect PLA2s, which we didn't detect at the transcriptomic level. And then onto South Africa, this is where it started to look very different from its transcriptome. So it's actually quite low in SVMP proteins, which we wouldn't expect from its transcriptome. It has a high expression of C-type lectins. And again, we found PLA2 and serine proteases here. And finally, for the Tanzanian individuals, we see this huge expression of SVMPs, which we would expect from their transcriptome. It was highly enriched in these transcripts. And again, a strong expression of C-type lectins, which we're seeing across all three. And you will also note at the end of all these spectra, you get this huge spike of adenosine, which we have done others have done research in our group. They think it could be to do with the predation of these species. So having kind of got an idea of what toxins are present, we then went on to look at if these are functionally active. So I picked to start with the snake venom metalloproteases, as these are a huge medically relevant toxin group within the viperids. And we have a fluorescent in-house assay at CSRI that we can use. So here, as we'd expect, there's the highest activity is from the Tanzanian venoms. Again, they're rich in SVMPs at transcriptome and proteome level, followed by the Nigerian. And again, I didn't expect from the transcriptome, but looking at the gel filtration, there's very low activity from the South African venom. So moving forward, we then looked at PLA2s, which again are another very pathologically relevant toxin. So all three of them had kind of comparable levels when we looked at gel filtration, but looking at the activity, there's clearly a lot more going on. We had significantly higher levels of activity from the Nigerian individual and lower from the Tanzanian and South African. But PLA2s are a lot harder to get a grasp on what's happening. You get lots of different isoforms of these toxins, and you can get catalytically active and inactive isoforms. So I think whilst this is only looking at the catalytic active data, there's some more picking to be done at this, but it was still really interesting to see that there's this variation starting. And then finally, I ran a plasma clotting assay, which can give us slightly more physiologically relevant data as to what might happen in bite victims, but it's harder to unpick what toxins are driving this because it could be a whole range of different toxins within the venom. And this is where we started to see really huge extensive variation between these three different regions. So you see when we incubate bovine plasma with the Nigerian venom, you get a significant increase in clotting rate. It's strongly coagulant. The Tanzanians seem to have a very minor increase in clotting rate. It's not significant. The spectra looked pretty much on what we'd expect for normal plasma under standard conditions. And the big surprise was the South African venom. So it was hugely anticoagulant. This assay runs for over an hour. By the time I'd taken the plate out, that plasma still hadn't clotted. So something is going on within the South African venom that's really interfering with these processes. So after kind of gathering a bit more information on what's happening functionally, we wanted to answer the question, is this venom variation going to have an impact on the efficacy of antivenoms used across sub-Saharan Africa? So for this, we picked 4 relevant antivenoms that all have Betis within their immunization mixture, or they are used in envenomations by these species. So Equitab Plus, which is using Nigerian venom, so used in West Africa, Sema polyvalent, Favafrique, and the Inocert Pan-African therapeutic. And I used a Lycor system to run some Western blots. And the beauty of this system is you can quantify the signal intensities of the toxin recognition and then compare these via a bar chart as shown on the screen. And what we can see straight away is Sema and Equitab Plus have the highest signal intensities against these 3 venoms, and they're both showing a slight preference to antivenoms corresponding to where they're produced. So Sema polyvalent is produced in South Africa, and it has a slight increase in its recognition of South African venom. And as I mentioned previously, Equitab Plus is produced using Nigerian Betis venom, and there is a slight preference here. But in all of them, you can see there is variation in the intensity of these antivenoms recognizing the toxins, but you can't interpret how they're going to function in an actual clinical envenoming at this level with just looking at protein recognition. So to start looking at this a bit deeper, we then applied these antivenoms into one of our in vitro assays, the SVMP assay, which I showed you earlier. So we incubated the venoms with 150 micrograms of antivenom for half an hour, and then assayed them to see what the residual SVMP activity is. And again, both SAMA polyvalent and Equitab Plus produce very strong, consistent inhibition of SVMP activity. Again, you see the strongest inhibition against South African venom using SAMA polyvalent. We didn't get the same picture with Equitab Plus. It also has a strong inhibition against South African venom, rather than Nigeria, which I was kind of expecting based on the Western blots. And again, you see a more variable picture with South African Inasrp. Both of them had one venom which they were not able to produce a significant reduction in activity in. I will note that obviously, given the lack of production of Fabapherique, it was quite an old vial I was using, so that could also be a factor in why we're seeing this. But again, you can see there is some differences going on between these venoms, but there's a lot more that we need to unpick here. So as a summary, what we've shown is that the three beta-sarotans from Nigeria, Tanzania, and South Africa do show a variation at their transcriptomics level. And then this variation is reflected at the protein level when we looked at both gel filtration and their functional activity. So we saw that with Nigerian beta-sarotans, it's this strongly coagulant venom that is rich in PLA2 and SVMP activity, which is then contrasted against the South African venom, which seems to be highly anticoagulant, but it's quite low, but it's a lot lower in SVMP and PLA2 activity compared to the other two. And then finally, the Tanzanian was very rich in SVMPs, but it had this minor procoagulant effect when we incubated it with plasma. And then it's a lot harder to sort of interpret what this means for the antivenoms. We've only tested it in a couple of assays, and neither of those can be predictive of clinical performance. We'd have to move to an in vivo model before we could make any of those assumptions. But in the assays we did test it in, SAMA polyvalent and Equitab Plus performed very well and very consistently. And we saw a more sort of changeable picture with Inasrp and Fabapherique, but they both did produce significant results against some of the venoms we tested. So where would we like to go further with this? I think proteomics would be hugely valuable. It would enable us to link our transcriptomic profiles with what's actually present in the venom, so we can start seeing what is happening to these proteins as they're produced. There's additional in vitro functional assays I'd like to run so we can get an even clearer picture of the activity profiles of these venoms, especially with the South African venom. I'd be really interested to sort of start finding out what is causing this hugely anticoagulant effect. And then, as I said, to understand the consequences of this variation and what it means for clinical therapeutics, we need to progress to a mouse model of either lethality or things like hemorrhage and necrosis, which are commonly seen as a result of bites by beta-sarotans. And then entirely separate to this, there are sort of unverified reports of patients presenting with different pathology having been bitten by beta-sarotans. So it would be a huge undertaking, but to gather multi-locational studies on the clinical presentations of these bites would be hugely valuable. If patients are presenting with different envenoming syndromes, it's incredibly valuable for medics to understand this. But what remains incredibly clear is that there's a lot to be explored with beta-sarotans. There's a lot more that we need to understand about these snakes. So I would like to finish off with thanking my two supervisors, Professor Rob Harrison and Nick Casewell, and then all of my wonderful colleagues at the Centre for Snakebite Research and Intervention, who I could not have got half of this data without. They've been brilliant. And thank you very much for listening, and I will be happy to take any questions.